All right. Thanks a lot for inviting me. Been a very interesting day so far. Promise to try to be fairly brief because I'm standing between you and coffee. And I thought about George invited me to speak and talk a bit about biology. I'm a bioinformatician, you might say, sort of like AstroInformatics, but with a bio in front. And I thought what I'd do, since it's a fairly short talk, is try to talk about one thing rather than my entire field. I thought I'd start with what astroinformatics seems like from the perspective of bioinformaticians, and it sort of seems like astronomers, a lot of what they do is Gaussian fitting, which maybe is astroinformatics circa 1960s, where you fit, do elliptical Gaussian fits to find the center of a set of observations. And maximum flux density. So, this kind of thing is very classical. And my biology colleagues apply these sort of techniques nowadays. And I just wanted to say that up front. So this is actually an interesting paper I thought I'd mention because it's local. It's by Long Kai, one of our distinguished colleagues in biology. He does an experiment called FISH. He has developed it into a very parallelized assay. FISH stands for fluorescent in situ hybridization, and so long, it's a bit hard to see on this image, but he obtains images of molecules and has problems that are very similar in nature to what people might do in Gaussian fitting. But that kind of thing is not really maybe where the cutting edge today is in astroinformatics, and we've seen lots of deep learning and other things, but I thought I'd pick a different topic at their intersection, and I looked, in preparing this talk, I'd try to figure out what does lie really in the intersection, and I found that there's one sort of technique which is somewhat at the intersection. I found a number of astronomy papers using this tool, and I thought I'd talk about it because in my field, this is one of the most popular things to do now, it and its many variants. And in some ways, I think it's sort of an interesting tool. I'll say in a minute what t-SNE is. It's a kind of a dimensionality reduction technique that is used as an exploratory data analysis tool, which in biology is very important, and I maybe will argue that useful in astronomy and where I think there can be interaction going the other direction, actually, from biology to astronomy. So I'll try to make my case in 15 or 20 minutes. First, let me say what t-SNE is. So this is an algorithm that was developed in around 10 years ago. And it's a really kind of simple idea. So it's designed to replace methods such as PCA, which are project data in a linear map. And in this specific method is based on the idea of fitting Gaussians to points in an ambient high-dimensional space, and then to do a sort of numerical optimization to find a two-dimensional embedding of the points that is faithful to a metric learned from these Gaussians in high dimension. That's a high-level description of what it is. I won't go into more detail here now. But I will say that although the technique is sort of heuristic in a way, it has had some theory developed for it after the fact. There's a nice theorem by Linderman and Steinerberger from Yale. These are applied mathematicians for Yale that argue that there are sets of parameters, first of all, that ensure convergence of this algorithm, whatever it is, and that moreover, it produces, if data is separated by symmetric in the ambient high-dimensional space, then the low-dimensional embedding will be sort of represent that clustering, and so it is actually a tool for clustering that has some theoretical guarantees. In particular, it's similar to spectral clustering. And this theorem is actually cited in the astronomy literature. So I thought I'd give one example to start off with. This is from a paper from 2 years ago where this was applied to data from the Gallus survey, and this is where I really am out of my league, so I'll stop before I dig a hole that's too deep. And I won't really say anything too much about this data or this paper because I'm not an astronomer. But each point here is embedded in 2D from 13,600 dimensions. And so what was done is these are spectra that were binned in 13,600 dimensions. This algorithm was applied. And the number of points here is fairly large. It's about, I think, 209,533. This is on the scale of what exactly we're at in biology. In our data sets now, we may have more points. I'll tell you a little bit later. Similar number. And we often operate around 20,000 dimensions if we're talking about human data, where we apply this technique, although we might also be in 13. This number, 13,600, is about the number of genes in the fruit fly. So one of the interesting things I'll say in a minute is this picture. It's supposed to sort of show you some kind of clustering of the stars by their type. And it looks a little bit fuzzy. It's not as clean as this picture, which is from another paper, with a lot fewer points. But here, the actual ambient dimension is 13. The spectra have been reduced to elements first. And that's not a coincidence. So biologists have figured this out, that it's not good to perform t-SNE on very high dimension. It's better to first lower the dimension. I'll show you in a bit how we do it. This is from another survey, the HARPS GTO survey from the local solar neighborhood. And again, this is sort of shown as a visualization. I kind of like this paper because for two reasons. First, it has these sort of pictures which really look like pictures from a biology paper. So here, the stars have been painted by which elements they are enriched for. So that's a kind of thing we do a lot with element replaced by gene. The other thing is that I really like about this paper is there's something for the biology community to learn. So there's a nice kind of analysis here of what the uncertainty in the measurements may do to this kind of t-SNE method. And that's something that biologists talk a lot about. Our measurements are also inaccurate, but nobody does anything about it. So here they did some analysis, which, again, I won't discuss in too much detail. So we do this also in biology, and that's what I want to talk about in my remaining time. But the first question I would like to sort of answer is, so what? So we also do this. But what does biology maybe have to contribute to thinking about astronomical data? So I'm going to show you a couple of data sets. I'll talk about this one in a little bit. This is my own data. Mine as in collaboratively made with the Anderson Lab and two of our postdocs, Brady Weisport and Jay Scaring, are here on campus. This is data from a jellyfish. I'll say more about it in a minute, but one thing I wanted to say that's already interesting, I think, is that in biology, this kind of data is producible locally by a biologist. David Anderson is one of our neuroscientists. It's the kind of thing that dozens, maybe hundreds of biology labs are producing now. It's a lot, the scale, not in terms of the number of points, but in terms of the number of data sets, is much larger because it's the kind of experiment you don't need a large sky survey to produce. Essentially, every lab has its own mini telescope, if you will. So let me show you a little bit what a modern paper looks like. And I pulled out a figure from a paper. It's not mine. It was published literally 2 or 3 weeks ago. It's a data set of the retina, cells from the retina in the eye from a mouse. So I really like this paper because first they did a very nice job making their data available for reanalysis and examination. And secondly, I think it's sort of a nice survey to give you. So this is one of the main figures, and it's got a lot of panels. And I thought I'd walk you through it a little bit and explain to you what it is about this data that's interesting and has maybe parallels that one could look at in astronomy. So first of all, this data set was collected over a period of time. So this is really a time series. And you can see this in plot B, I think, if you look up to just to the right of plot A. That's a t-SNE. And the plots are colored by time. The points are colored by time. Now, what is each of these points? So in this experiment, each of these dots is one single cell. And there have been 20,000 measurements made in this one cell, namely, for each gene, how much of the gene is expressed. That is, how many RNA molecules are we seeing for that gene. So that gives a probability distribution over the genes, of which there are about 20,000. So instead of 13,600, the ambient dimension is 20,000, and each of these points is a single cell. In this experiment, they looked in aggregate at about, I think, on 100,000 plus cells. So it's in the same order of magnitude as the Gaussian survey, the data set that you saw. So first of all, there's a temporal order to the data. So the cells have been collected from different differentiation points in the mouse as its retina is developing. So that's the first thing I wanted to say. And the second thing is on the right hand, on plot C, are the cells that have been clustered. So this is the analogy to the different types of stars. And what you see below that in D is looking at which genes are characteristic of the different types of clusters. So that's very similar to what you just saw before. One of the things in biology is that we have a lot of knowledge in these sort of systems about what the cell types are already. We know from other orthogonal experiments. So when it's producing data like these, there's a real sort of ground truth to look at. Now, E and F are similar to the t-SNE plot, but they are what's called a UMAP plot, which is very, very new kind of method. It's basically another kind of embedding, different from t-SNE. That's sort of a differential geometric approach, if you like, making the assumptions that the points are uniformly distributed on a Riemann manifold. And again, I won't say too much about this method for lack of time, but it produces different kinds of embeddings, which have also a theory behind them and other nice properties, and may be more or less suitable to the kind of data you're looking at. So, as I said before, plots D and also G below are the kind of mapping where you're replacing chemical or spectrum frequency by gene. Now, one of the things that biologists have played around with a lot, much more than I think I can see in the astronomy literature in the papers I've seen that use this technique, is really examine what's the best workflow for actually producing good analyses. And this is a software tool that's very popular in our field. It's called Seurat. It's from a guy at the New York Genome Center. And it's written in R. Again, the favorite statistical toolbox of choice for biologists. And down here below, you can see sort of what the workflow looks like. So you load in the data, and the first step is to do a normalization, which is also done on the spectra. But then there's a step that's crucial, which is to find the most variable genes and to perform a PCA first, not to 2 or 3 dimensions, but to 50 or 100 dimensions, basically to denoise the data. So that PCA is used as a noise. And I can tell already by just looking at these plots, this the first one I showed you from the Gaussian survey did not do this, and I don't think. So that's why it looks sort of more fuzzy-like. And the clustering is typically done in 50 dimensions prior to doing the t-SNE, not after. And so all of these sort of approaches have been investigated, and I'll say more about them in a second, about all the different flavors and variants and research that's gone into all those things. Now, one of the things people do, which I think is quite interesting, is with these sort of data, is they create what are called pseudo-time plots. And this is a figure from Cole Trapnell, a former student of mine who's at the University of Washington, giving a rationale for biologists why they should do single-cell experiments. And I won't talk about A and B. These are basically the rationales to avoid Simpson's paradox. But I want to talk about C. One of the things is that even though we do collect time series, the time in biological experiments is often not very accurate because cells are in states that are very difficult to synchronize sometimes. And so the idea in pseudotime is to order the cells according to their profiles in this case their gene expression profiles, in a way that you may actually think would be very sensible to do with spectra. So you might imagine taking stars and ordering them directly from their spectra, and people do that also, but this is done in the context of t-SNE and these dimensionality reduction approaches. So I'll show you, this is a figure from the same paper I just showed, where they're doing this exact technique, performing it for these retinal cells. So down here, you don't need to be able to read the gene names too much, but these are individual genes that have been selected because they are varying highly on these trajectories of the cells as they've been ordered by pseudotime. So, this particular gene is going up, and this is going up and down, and what you can see on the right-hand side is the ordering of the cells that's been done automatically. And this is basically, there's a number of ways of doing this. It's kind of like a traveling salesman problem among the cells to find, the shortest tour that will take you from one to the next. There's a very extensive literature now on how to, to do this pseudotime in the best way. Really lots and lots of techniques to do that. So let me just tell you in a few minutes what kind of experiments are possible now and why we have so much of this kind of data and why we're exploring it in such detail in my field. So this is a picture of a jellyfish called Clydia hemispherica, and it's a jellyfish that I didn't have particularly much interest in or knowledge about until I came to Caltech about 2 years ago. I was introduced to it through my collaborator David Anderson, who's interested to study this jellyfish in order to study behavior. And one of the interesting things about an animal like this is that nowadays, performing a single-cell experiment on it, that is to say, an experiment in which we're going to measure the expression profile in each and every cell to produce a few tens of thousands of data points, is an experiment that runs in the order of 2 to 3 weeks and is not very expensive, order of maybe $5,000, maybe $10,000 if you want more data to perform the experiment. So we did an experiment actually comparing conditions where we feed and starve a jellyfish. I'll mention this as well, another area where this biologists are quite advanced, because one wants to do experiments and not just one static measurement. There's been a lot of thought about how to merge data sets. And one of the ways to do that actually is by using autoencoders and other neural net techniques. But in any case, in this particular experiment, we did 2 different conditions and many individuals in each condition, and we have a technique we developed in my lab for labeling them. I won't talk about the actual technology. There's a lot of biochemistry and enzymatics and sequencing technology and all sorts of technology, microfluidics, that goes into getting the matrices out. But it doesn't take very long to get the matrices and to go to this kind of picture. This particular picture, the dots are colored by the different individuals, and it's sort of a nice picture because it tells us the messiness of it, the intermingling of the colors, tells us that we don't have a lot of batch effect in the experiment. I wanted to just show you these pictures, not because I want to talk very much about this experiment today, but I wanted to show you that we make exactly the kinds of pictures that you just saw a minute ago, except instead of highlighting which chemicals are present in which types of stars, we're seeing here which genes are active in which types of cells in the jellyfish. And these are various markers that are lighting up in different clusters that relate to different functions of this jellyfish. You're actually looking at a map of the entire animal. It doesn't look like the animal because it's in this weird t-SNE embedding, but really it's representative of what we think are all the major cell types of this animal. So one of the really, what we do basically nowadays in bioinformatics, a lot of the work is about delineating the cell types and following them along and trying to guess and learn and predict which genes are, which types of cells are precursor cells and which are differentiated cells, and which cells are changing from one to the other. And it's interesting. I saw that I looked at these, I mentioned these two astronomy papers. It's very much the same kind of thing is going on in those papers. The t-SNE, the clusters are being cells are being, not cells, confused, stars are being extracted by hand, and people are looking at, at which elements are in which and how they're changing. It's sort of the synthesis of the automatic approach and then going in and looking by hand. But again, I think we have a lot of very fancy tools now to do this kind of thing. And you can see that in this data set, we actually like UMAP projection, which tends to be very much better at in cases where there's some kind of change happening that's where the points are essentially lying on some kind of a trajectory. So this is the picture I showed you previously that I started with in the biology side. In our case, we don't have so we don't have the analogy of a spectra knowing which elements correspond to which spectra. So we have to basically annotate each of these cell types according to known databases of genes in other organisms that might be similar but aren't always, and we only have partial information about what those genes are doing. So in biology, the part of labeling these clusters is really the hard part right now, and really active area of research. It's what we spend a lot of our time doing, and linking together all the databases to do this is one of the endeavors that we do. So in terms of why I think there's some possible fruitful interaction that can happen is that in bioinformatics, been a lot of work looking at how to combine PSCA with t-SNE and other dimensionality reduction techniques, and what's the optimal method, and where is there theory to, where is there not theory. Really, a lot of different approaches have been looked at. The clustering methods have been examined in great detail, and I know that in this community also people work on that, but there's really been a tremendous amount of work on clustering and also linking it together with the dimensionality reduction techniques. A very big area that is of great interest is imputation. Again, I think could possibly be fruitful in the astronomy setting. And looking for markers, trying to define what really is a novel type, what are the differential markers, and lots of really sophisticated techniques to do that. In this setting, the data is count data, so that's a little bit different, but a lot of it's really like very, very similar. So I think I wanted to end by 3 o'clock. I'm almost out of time. I'll just conclude by saying that we're just across the hall over here. I know some of you are local, but many of you are not. But wherever you're coming from in your universities or your institutes, there are almost certainly bioinformaticians, many of whom actually come from physics or even astronomy. So there's even a sort of common educational background often. And I think there's really a lot of room for having what I can see as more interaction between the communities, which is one of the reasons I was happy to come today, and I look forward to this whole week. So thanks a lot.